Hi guys. So this this lecture is about the cognition and dementia. Um, so the context comes from the textbook, chapter 58, the cerebral cortex, and I also use some review article to enhance some more updated information about the dementia. So with this lecture, we will break down into these four areas. The cognition, we will describe what do people know about the cognition, how do we explain it, how do we make sense of it. The next is the cortical function about this cognition and the location in the cortex to work on this function. Following that, we will talk about the connection between the cerebral cortex, the neocortical region, and the thalamus. We will kind of we talked about each different connection earlier, but we didn't really systematically describe this very closely related cortical thalamus interaction. So we will use this part to summarize them and as well as enhance or rephrase or remind you guys that we learned earlier about this different region of the cortical thalamus interaction. And following that is we will talk about the injured disease condition to make it to reduce the cognition, and the major one is the dementia. Well, the major type is dementia, and we will mention this disease, Alzheimer's disease, as the major disease for the senior dementia. So what is cognition? So cognition by definition is the mental or brain or mental process to conduct thinking, meaning to think, right? To generate thought as well as awareness. So what does it mean? You know, when we talk about thinking, we know what we mean, right? But we talk about the awareness, we know what it means, but how do we break it down to more like direct neurological function? Thinking is considered a process of information. So, you know, to put information together and come out of some new idea or different direction. Thinking can be a mental process for motion, making decision, making plan. So that is the thinking. So in the way that thinking is to put this information together, what does it mean information? Information need to be acquired. So it will be something like knowledge, something you read, or experience, something you experienced before. And those are what you read, what you learned, what you experienced are something stored in the memory. So cognition or the thinking in a way is to gather, to search and put the memory, several pieces of memory information together. And that's the thinking. As well as for the awareness, awareness would be something that you sense. There are two area of the awareness. One is the external, that you are aware of what you are located. You are aware of your environment. So that is awareness of the external. So this is about sensory, right? You are aware of the environment because you see, you feel, you touch, you observe. So that's the sensory, as well as the awareness of yourself. You are aware of your own status, your own present. So that's awareness. So it's sensing the environment, as well as sensing yourself in the environment. So when we look into the breakdown of the cognition, we can see, let me do this one here. I hope you guys still can see the screen. I hope, okay. I don't want to take the chance. I will just close it. Okay, either way. All right, so when you see when you break down into different domain, the major domain of the cognition, you can see that is what it's about. Attention, because you are aware of the environment, that environment, we have so many information, so many sensory information, so many sensory signals in the environment. What capture your attention, right? So that is drawing our awareness. And sometimes some like mild difference of the attention reaction can make huge difference, like autism disease, right? That is how we respond to the attention. Some people are more sensitive to the signal, and then for most of us, we just treat those signal as random noise, so we don't get bothered by that. But it's not always true for everybody. Judgment. So this will be thinking, and then you make decision. So that is, you gather information, you collect the sensory signal, connecting you with your experience, with the knowledge you acquired, then you make decision. Social cognition, this is like another huge topic. That is how you are aware, or how you think of your presence in relationship with your society. So this social cognition is the ability to predict others' response to your own action, or to predict others' reaction based on the environment. So you can read facial, you can predict what people respond. So this is not just the brain, but also the experience you have with the environment, with the society. And so this is cultural related, that different culture may have different context, different components in that social cognition. And that culture basically is built up based on your experience in that community or in that society. Executive function, this is more like related to judgment. Judgment can be you decide right or wrong, or like it or not like it. Executive function will be based on your judgment, and you make a plan to achieve a goal. So this is not just the sensory, not just the memory, not just the experience, but also function, motor, right? Also the action. Intelligence will be also based on, what does it mean intelligence? What does it mean intelligence? intelligence means that the most obvious example would be a person is intelligent enough to make right reaction, right? So that is that you can make the most proper or most effective reaction in a certain condition. And how can you achieve that kind of intelligence? Experience, knowledge, thinking. You have experienced something, you learn something, and then you put those together and then make your own judgment and then make your own reaction based on those. And so intelligence is about memory. Experience is a memory, and so that leads to the next one, memory. There are different types of memory: short-term memory, long-term memory. We also can classify memory into more like skilled memory, is motor related, or something more declarative, meaning that you can describe it. Type of memory, these are very different kind of memory. And that memory, storing the memory as well as retrieving the memory from our brain is an important function of the cognition. So that is cognition. Most people believe that cognition is a function in the brain. It can be argued that it's not, but most people believe it is, and we believe that too. So if you look at the brain, right here is a brain, you can see that these functions not only can be performed in the brain, they are also performed in specific location of the cerebral cortex. We kind of know different location of the brain already. Excuse me. Different location of brain already, and very roughly we have the prefrontal cortex, the frontal, the parietal, the occipital, the temporal lobe. And so that's different location, different lobes of brain. And we kind of collectively already described different region. We put a lot of effort talking about the somatic sensory, talking about the visual cortex, talk about the motor. Now we kind of like go over everything and describe. You can see that all these functions are in the brain. For example, in the prefrontal cortex, this is the region known to perform the function for higher intelligence. It's basically, you can make reaction, or you can make a good summary based on the experience as well as the environment. So this is important here. Decision making. However, you need to know that this region is very closely related to the limbic system. So our decision making, our judgment, our executive function are all controlled by here. And this is very closely related to our emotion, the limbic system. So our decision making is, if you know the neuroscience, you should know that our decision making is not totally rational. Our decision making is very emotional driven. Okay, your reward system, your fear, control or affect your decision making because they are closely related. The prefrontal cortex region, sorry, the the frontal region is somewhere we can see that we have the motor here. We also have the voluntary eye movement. We talk about that. What does that mean? How does you connect this one with all this domain of cognition? This one is closely related to the attention. So attention is something you respond to the sensory, and that drive our eye movement. So we have very close like connection between the occipital and the frontal uh, uh, region, and that drive our voluntary eye movement. We also have this region. This is broadcast area. That is how we use our motor function to conduct speech. This one is also closely related to the Wernicke area. This is another speech center that is more like sensory spectrum of the speech. You understand reading, you understand the language, you understand what people are talking about. You convert that auditory signal or visual signal, as well as tactical signal, into a sequence of the words and then make sense of it, understand the syntax of that language. So that's the Wernicke area. Then here we have the sensory motor, and then sensory, you detect the information. This parietal region. Parietal region connect all different sensory signal together and then make sense of it. Extract the feature of that sensory, integrate that sensory signal. The most beautiful area of that function is the language. So this make us smart. This make us intelligent because we are probably the reason why we are human, compared to other animal, is that we not only have the language, we can generate that language function from collectively different type of sensory mode. We can read, we can listen, and we can collectively or use all this information to generate a language. Now it's vision. Here is the temporary region. We have the some part of it is for the auditory, some part of it in the inferior part of that temporary lobe is the memory. We have one region very important is hippocampus in the inferior part of the temporary lobe. That is the important region of the memory. So you can see that all these function, even though it sounds very like mental, it seems very like mental function, they are neuroscience function, they are neurological function, and that they are conducted in the brain. So that's the cognition, and that is the functional region, functional area in the brain. I like this picture too. In addition to this one will show you like lateral part of the brain cortex, but you need to know that brain are folded in different way. A lot of the function can be described in the cortex, but we also have several subcortical region. So from this one, you can see the lateral side of the brain, as well as the middle side of the brain right here. From the middle side, you also see the cingulate cortex. You also see here is the orbital prefrontal region, and you kind of can start to appreciate how close the limbic system to the prefrontal region, and that I hope that provide you very visually um, noticeable connection of how our emotion can influence our decision making. Yeah, and a lot of you already know, through our um, earlier lecture, or through your other course, you already know that we know that there is there is a reward system here in this limbic system, and that whether you like it or not, I mean, whether based on your experience, that you make decision based on your experience, that you like it or you are afraid of it. So based on that, the reward, dopamine, or the fear, amygdala signal, that drive our decision making, the frontal cortex. So that's that. Now, another thing I want to bring is that now we know different cortical region provide different function, and collectively they allow us to have the mental process for thinking, for the awareness, for the attention, for the cognition. I want to bring another thing that is also very important, is that in addition to this cortical area doesn't work alone. They are closely related to the another region that is thalamus to complete this function. So I want to talk about the cortical thalamus interaction, and that is called the thalamocortical system. This is the picture from the textbook. You can see that different cortical region, they don't work alone. They work through bidirectional connection with the thalamus. Thalamus basically located right here. So you can see that this center whole, like core, is the thalamus. So thalamus can interact with the cortex, and it's not unspecific. It is specific. Specific cortical region connect with specific hypothalamus. So that is very important, that our thalamus, although it's not cerebral cortex, however, the interaction between the specific connection of the cortex with the thalamus are important. If we cut any of these connection, we basically lost the function of the cerebral cortex. It doesn't really matter if your cortex still intact or not. If you lose that connection, the cerebral cortical function will be gone. So that's that important. And you can see that here is the region indicates that where the thalamus is directly associated with this specific region. We kind of went over the thalamus already. We know that there are two lobes of thalamus, and that the thalamus nuclei can be classified into anterior, ventral posterior, dorsal, lateral dorsal, or medial dorsal. This is the ventral posterior, and also the vulvar, as well as the two genicular nuclei, the lateral, medial, as well as that here, these lines are 1 meter, we call it laminar. In the laminar, we have several nuclei. Inside of the laminar, we call it intralaminar nuclei. We kind of went over that already. Now we need to know, okay, these cortical regions are closely related to specific thalamus area. For example, prefrontal are very tightly related to the dorsal medial nuclei. Dorsal medial right here, dorsal medial nuclei. The motor, prefrontal motor is related to a ventral lateral nuclei. The primary sensory cortex is related to the ventral posterior lateral nuclei right here. The parietal are related to the lateral posterior right here, and the occipital, this is about the vision. It's part of it is very important to interact with the pulvinar. The visual directly is related to the lateral geniculate. Here is the auditory. Auditory is related to the medial geniculate body or nuclei. So you can see that all these, each of these directly connect with the thalamus. So we kind of like touch on some of it. Perhaps we already touched on all of these. Doesn't matter. And here with this part of this lecture, I want to summarize, systematically describe all these different nuclei and their relationship with the cerebral cortex. This could be this is important that you know that thalamocortical connection and that interaction is critical to maintain or to provide in a way that our awareness, the level of our brain activity, the level of our clarity of our thinking, that's that important, okay? Here is showing you that if the connection, any of the connection is cut, then the brain function is not functional. It's usually not happen. You know, we don't really cut it. Nobody would like to cut it. This usually happens when there's injury. However, something happens every day to us, which is sleeping. So this is the region basically suppress all the signal into the brain. And that is the time that we are asleep. So that's that important. This is a region to in a way to adjust the level of our clarity of how our brain functions. So that's that. All right. So basically, as we mentioned earlier, that the thalamus is like the hub to the cerebral cortex. There are different nucleus, and they almost except the olfactory, the smelling. Other aspects of that olfactory only enter into the olfactory ball and located in the limbic system, right here. So this is the olfactory ball. You can see that this is not the visual, okay? Don't be confused. This is the olfactory ball. And then underneath this one is the nasal cavity. Cranial nerve 1 enter into this region, and several like cortical region underneath here process that sensory of the smelling. So except that other will go into the thalamus before they enter into the cerebral cortex. And so this is the like the hub of the brain. We used this picture earlier, kind of frame it up, and this basically summarizes everything we are going to talk about today. Majority of those. So we talk about this is our old friend ventral posterior region that is related to the somatic sensory. So the DCML signal from the tactile, from the skin will all go in here, ventral posterior region. We also talk, when we talk about the visual system, we learned that the retinal, optic nerve, and optic tract project directly into the lateral geniculate body or lateral geniculate nuclei. And then project into the V1, the primary visual cortex. Let's go from this one is also very, another like hanging out there, medial geniculate body. This is the inferior, this is from the inferior colliculus. It's a process, it's a signal from the auditory signal. And that is the process is to, get the information for our auditory signal. VL. What is VL? VL is from the cerebellum. So I think it was just a week ago that we learned about the cerebellum function, and the cerebellum has several deep nuclei to provide the output signal. One of those are sending into the brain. That is the dentate nucleus, right? Dentate, I think that's dentate. And so that sends signal into the cerebral cortex, into the brain, need to relay on the thalamus, and that's where it is. VL, ventrolateral. And because this is about the motor, cerebellum control our balance, body balance, so that sends signal into the motor cortex. The earlier as part of the VL, the earlier or anterior VL, as well as the total VA, are associated with the basal ganglia. So again, about a week ago, that we learned basal ganglia function is to channel through our decision making and make a movement. And that decision making through the basal ganglia, the signal basal ganglia will need to go through the thalamus before going back to the cortex. And that thalamic nuclei to process the basal ganglia region is the part of VL as well as VA. You can see that it says it's from globus pallidus. So basically in the basal ganglia, it goes through different parts. We have the direct pathway and indirect pathway, but the output of that process is either substantia nigra as well as the inner segment of the globus pallidus. So that's that. And that output signal will go through the thalamus before going to the brain. Not directly the motor, but the premotor, because that is a channel through, say, yes, you can do it. It's a good time to do it. And so the premotor will capture that information and then to decide the sequence of the movement, then tell the primary motor cortex to do it. So we basically go over that. And there are some other region we have not really touched on here. Anterior, who do they connect with? Okay, anterior. Here, the medial dorsal, who do they connect with? Here, a little bit, right? This region, lateral dorsal, who do they connect with? This one, Pulvinar is visual. We kind of touch on that, visual and auditory. So basically, these two will, part of it will send. They don't directly send to here though, but this signal, this like the type of the information they are processing are similar. So we will touch on this extra region. We also touch on the intralaminar. Remember what is intralaminar for? Intralaminar is for slow pain. So slow pain signal sending here. Intralaminar is part of the ascending reticular arousal system, and is part of the ascending reticular arousal is part of the reticular formation of the brainstem. So those regions process the pain signal. So this is important to the pain. One more region we also did not talk about yet is these part. This is another group of nuclei, reticular nuclei. Don't get confused with the reticular formation in the brainstem. This one is reticular nuclei. It's located in the border of the thalamus. What does it do? It inhibits the signal of the thalamus. It suppresses it. So this one sends signal. If this signal is very strong, it will shut down our brain. So this is the like break of our brain activity. This one sends inhibitory signal into each of these thalamus to suppress it. During like a deep sleep, we sleep has 4 different phases, different stages. During that deep sleep, REM or non-REM, if you don't remember, don't worry about that. These reticular nuclei will send a strong like spindle wave. That wave propagate directly into the thalamus to suppress the thalamus, and that basically shut down our brain connection with the world. Brain can still think, can still kind of like dreaming because all this thinking still circulating in the brain, but not through the thalamus. And also sensory signal cannot pass through the thalamus to the brain. Basically, it shut it down. So that's that. Very interesting stuff. All right, so that's what we are going to do. Ventral posterior lateral, we talked about that already. This is somatic sensory relay. The somatic sensory go through DCML and the ALS. The ALS has 2 parts. One is the neo, the other one is paleo spinal thalamic tract. ALS process signal of the pain. Medial lemniscule DCML, dorsal column medial lemniscule system, process the tactile signal. And all this will go through the ventral posterior lateral, ventral posterior lateral nucleus before they send into the primary sensory cortex. Another is the ventral posterior medial. What is that? It's right, I think it is ventral posterior medial, ventral posterior lateral. This one doesn't really show it very clear, but you can see it basically this one here. It's called the arcuate, but it should be, just remove that one. It should be the ventral medial. Yeah, so I can show you, maybe I have another picture here. This one is better. Ventral posterior medial. All right. Supposed to do it here. Yeah. Ventral posterior medial. So that makes more sense. That's that one. Yeah. So ventral posterior medial is part of the, is related, is very near the ventral posterior. So this is also processing the sensory, the somatic sensory, but these medial part get information from the trigeminal nerve. So this is sensory of our facial, because our facial sensory doesn't go through the DCML, doesn't go, it goes through the trigeminal nerve, the cranial nerve 5 into the brain. So that's that ventral posterior medial. So collectively, these ventral posterior lateral ventral posterior medial are collectively called the ventral basal complex. And what's in common between these two? They process basically the somatic sensory cortex, and they project into the primary somatic sensory cortex. That's that. Here is just showing you that the trigeminal nerve, we have 3 branches of the trigeminal nerve. They gather the sensory information from our facial, our mouth, our tongue, our head, basically, into the brain. And before they reach into the, topological matching sensory signal in the somatic, in the primary somatic sensory cortex, it has to go through the thalamus, and it goes through the ventral posterior medial lobe, or nucleus, ventral posterior medial nucleus of the thalamus before going to the brain. Intralamina is also something we talk about. This is the region process the pain signal. Intralamina has several nuclei. The largest one is called the centrum medianum. And all these gather the pain signal. As we talked earlier, that there are 2 types of pain, fast pain and the slow pain. So what goes through this intralamina? The slow pain, okay? The fast pain will go through the ventral posterior. Fast pain are only in the skin. We can identify the location of the pain. Slow pain comes from the skin as well as the visceral. When it's visceral, we basically has no idea where it is. But it's painful, right? And so that goes through here. This one is also part of the ascending reticular activating system or arousal system that include the intralaminar as well as the reticular formation of the brainstem. So this is a signal to process the pain signal. When they got triggered, they basically will increase the alertness of our brain function. But how do we do it? How do the brain increase the alertness of the, or the awareness of that signal level? It excites the thalamus. So they interact with the thalamus in order to increase that activity level. And also they send signal into the cortex to tell the brain that there is something that we need to get rid of that, the source of that pain. So talking about this pain, let me summarize all this pain signal in the higher brain region. That is the pain include the fast pain and the slow pain. So they are different, okay? Don't just, I'm faster or slower. It's not just the spectrum. It's different pain. Fast pain goes through the A delta. fiber is and go through the spinal thalamic tract. This one this is the fast pan. This is the fast pan. They will go into the ventral basal complex, including the ventral posterior, lateral, or the ventral posterior medial. So that's the fast pan. However, the slow pan will go majority step innervated in the reticular formation, but part of it will go into the intralaminar nuclei. So the destination of these two in the thalamus are different. They innervate into different nuclei in the thalamus, and that intralaminar nuclei is part of the ascending reticular activating system, and that is part of the very important components to trigger this very painful slow pain. So that's that. Ventrolateral is part of the thalamus. It receives signal from the thalamus. But we know that from the thalamus, it has three output nuclei: dentate, interpost, fastigial. The dentate is one going into the cerebral cortex. And before they go into a cerebral cortex, they have to go through the thalamus. And the nuclei to take that information is the ventrolateral nuclei. Where is that? Okay, so that's that. Okay, good, good. So that's that. Okay, so this is not entirely new. We learned it already. This just put them together. Know their relationship with other thalamic nuclei. As for the, so this is ventrolateral, but more like posterior part of ventrolateral. How about the more anterior part of the ventrolateral? That as well as the let me show you this. That's right here. Ventrolateral. So this is more posterior part of ventrolateral, is to receive signal from the cerebellum. How about the more anterior as well as the ventral anterior region? These region receive signal from the basal ganglia. And so that's this one here. Thalamus, basal ganglia. This is basal ganglia. Basal ganglia, we have the direct pathway. That's this one here. Striatum go through globus pallidus internal segment, or striatum to the substantia nigra, pars reticulata. These two will then go back to the brain, and before they get back to the brain, they go through the thalamus. Which thalamus? The ventral anterior, and the anterior ventrolateral. So that's that. Either direct or indirect, these two components are the added part to make it indirect. Either way, in the end, they will all go through the globus pallidus internal segment as well as the substantia nigra. These goes back to channel through all the different thinking, channel through, filter out what's inappropriate, and then allow the brain to make the reaction. That signal will go through the thalamus first, and that thalamus region is the ventral anterior as well as the part of the ventrolateral. Now we kind of go from ventral posterior, that's what we most familiar with, to further anterior, ventrolateral, ventral anterior, now the anterior. What is anterior for? Anterior here is more like emotion related. It's very related to the limbic system. This anterior is very related, highly related with the limbic system. In the limbic system, the emotional process, there is one important circuit. It's called the peptide circuit. These peptide circuits start at the hippocampal formation. This is a region, is output of the hippocampus. It's also called the subiculum cortex, subiculum cortex. Their hippocampus has a fiber called the fornix. This fornix will go over to the front, connected with the mammillary nuclei. This mammillary nuclei will then have the tract into the thalamus. Through the thalamus to the cortex, but the limbic system, the limbic cortical region is in the singular gyro, the cingulate cortex. This cingulate cortex then going through the cingulate cortex back to the entorhinal cortex para. So this going to the temporal lobe, the inferior part of the temporal lobe, to the entorhinal cortex, back to the hypothalamus. So hypothalamus is where we have experience. The experience will provide information about good memory, bad memory. So it drives our emotion when we receive new input. This new input, whether there's a visual, there's auditory, there's a sensory, will send into will match with our memory, and to classify it, to kind of make sense of what we experience, and that will go through this channel. So this is the peptide circuits is the core circuit to process limbic information, the emotional information. And as you can see, although this limbic information doesn't really directly contact with our neocortical region, but it contact with our subcortical region, that is the singular gyrus. You probably saw that singular gyrus in the very beginning right here. If you forgot, it's right here. That's here. So this is neocortex. This is a singular gyrus. And the peptides from the mammillary, this is the fornix. Mammillary will go through the thalamus. Which thalamus? The anterior nuclei. Then go into the singular gyrus. So that's that. So that is what we do with this anterior nucleus. The anterior nucleus is part of that peptide circuit, is part of that limbic system. So that's this one, anterior nuclei. So that's that anterior thalamic nuclei. So part of this limbic. Basically, all these are in the limbic system. Yeah. All right. So this one just showing you, this nuclei that we just mentioned about. Here is the, this kind of that view is kind of integrate from the temporal lobe as well as from the medial lobe. So if you want, you can use, more like a 3D image to look at that. But this one give you the idea, where are we, what are these components we talk about. So basically what you show here are all very basic, or very like emotional related nuclei. And you can see that it's very close to the prefrontal. That's where we make decision making. As for the peptide circuits, it's from the hippocampus through this fornix. Fornix is 5 meter. fiber send signal into the mammillary, then send to the singular, but before they send into the singular, they send through the thalamus, the anterior portion, anterior nucleus of thalamus, and then send to the cingulate cortex, right here, cingulate cortex. Cingulate cortex will then send back into the hypothalamus. So that's that. So that's the anterior. All right. So what else? What do we have? Yeah, let's go back to this one here. So what do we have? So we have the ventral posterior, more like sensory, ventrolateral, ventral anterior. This ventrolateral receives signal from the cerebellum. What's the cerebellum for? Keep our body balance. So this drive signal into the motor. Ventral anterior receives signal from the basal ganglia. What's the basal ganglia for? Again, channel all these thoughts and make a movement decision, a decision to make a movement, make reaction into the environment. So that these two send signal into the motor cortex. What else? And then anterior, this is more emotional related. And then go to here. This is the medial dorsal region. These regions are related to the prefrontal. So these are more emotional, then gradually move into the decision making. Prefrontal. So medial dorsal is closely related to the prefrontal. So that's this one here. Medial dorsal thalamus receive lots of limbic information as well as the prefrontal cortex. They send out signal into two branches. One is the prefrontal, and that will make the decision making, right? Make the judgment, make the planning. Make the planning executive function. So our executive function, even though that to a lot of like social economical scientists, I think one of those kind of new Nobel Prize winner, the idea they win it because that they found people make decision unrational, not based on rational, and then that's how they find an economic like a mechanism to describe why people make decision non-rational, and it to the social economical scientists it seems like a novel idea, but for the physiology or for the neuroscience people it makes sense, because the connection tell us that our executive function is non-rational. It's very heavily affected by our emotion, by our limbic system, and so that's that. Another output from the dorsal medial or medial dorsal thalamic region is to the hypothalamus. So collectively, they the signal from the limbic system, our emotion, can affect our heart rate, our breathing, our eating, our hungry, our thirst. That's all controlled by the hypothalamus. So the limbic system modulates hypothalamus through the medial dorsal thalamic nuclei. All right, so that's medial. Another part is lateral posterior. Lateral posterior is related to the parietal. This parietal is a associative region of the sensory, and so these lateral posterior thalamic nucleus receive the signal from parietal and then project into the motor in order to provide an integration of the sensory motor signal. So that's that. So I think we are kind of circulating from the anterior back to the, so from the ventral posterior, ventrolateral, ventral anterior. Anterior, this is the limbic system, pelvic circuit, medial dorsal, we talk about that. And then we are going to go through this one here, lateral dorsal. Lateral dorsal is about the parietal. Okay, we talked about that already. And then now we are going to talk about this one here. Lateral posterior, lateral dorsal. Now we are going to talk about this one here. Pulvinar. So pulvinar is the region to get information from both visual and visual and auditory. Lateral posterior. Lateral posterior is from the parietal. Okay. Pulvinar receive the visual and the auditory signal, and they don't directly receive signal from the visual directly or the auditory directly. They receive signal from the visual cortex as well as the superior colliculus cortex. So from the cortex, it's kind of like a reciprocal circuits that from the cortex into the thalamus, then back to the cortex. Auditory from the temporal lobe. The projection back to the parietal, more like inferior part of the parietal, as well as the adjacent temporal lobe. So this part receives sensory signal from the cortex and then back to the sensory integrative region of the cortex. So that's the pulvinar nuclei. Now we have the other two. So we just talk about the these region, and we don't really talk about lateral dorsal, but other than that, we kind of go over every part. We talk about the lateral posterior, and then these two. This is lateral geniculate. This is medial geniculate. Lateral geniculate is about the vision, visual signal, the vision, the visual field project into the retina will directly project into the lateral geniculate nuclei. The auditory and that will project from here, project into the visual cortex. We showed this earlier in our lecture. As for the auditory, we didn't really touch on the auditory because of time. If we have time, we will talk more about that. But auditory from the cochlea sense signal, in the end, it will need to go back to the brain, right? Auditory cortex. Before it reach to the auditory cortex, it goes to the medial geniculate nuclei. So this one summarize this auditory signal. The cochlea go into the brain, directly innervate into two nuclei. One is the cochlear nucleus, and then it will go into either the ipsilateral or the contralateral superior auditory nuclei. This tract will send into the inferior colliculus. I want to show you this picture. This is the Auditory signal, where is that? That's this one here. Vestibular cochlear signal, cranial nerve 8. When they innervate it, it's here. It's where 8 here. It's here. They will eventually send into the inferior colliculus. I want to show you this one here because I want to show you that this is the brainstem and these two like bumps on the back of our brainstem include these two bumps, or four, two sides, include the superior colliculus and inferior colliculus. And these are very interesting. These inferior process the auditory, superior process the vision. So as I mentioned, as we kind of went over that, but if not, I can show you here. This visual signal, the visual information, the majority going into electrogeniculate and that project into a visual cortex that provide us vision, but a copy of that information actually sent to the superior colliculus. That's right here, superior colliculus, this little bump here. And that provide our eye movement, provide our attention. So that's that one here. All right, so where's that? So inferior colliculus then sent into the thalamus. In the thalamus, it goes through the medial genicular nuclei, then sent into the brain. So that's that. So that. Okay, another very important one is the reticular nuclei. So that is reticular nuclei. This one doesn't send to the cortex, but this one sends signal. Here is thalamic reticular nuclei, TRN. This one sends inhibitory signal into the thalamus. So it suppresses the thalamic function. When it suppresses the thalamic function, it's basically turn down our brain activity. And as I mentioned before, it's very important to tune the active level in the brain, and it becomes very important, especially when we are asleep or awake. So this is very important nuclei to control our sleep or awake. Okay. So I know you guys, a lot of you guys are interested in the the sleep. A lot of you guys interested in the topic about the coffee, and a lot of neuroscience are fascinating. And when we talk about the sleep, there are many parts of our body system regulate sleeping. Caffeine is one of it. Thalamic reticular nuclei is also another one of it. It's very important one to tune our brain clearness or awareness or the level of activity. So that's that. All right. So here it's just showing you that this is the reticular nuclei and the day project. Another way to think about it is that all this not just one line, this basically wrap around the thalamus. So everything going into the thalamus will have to go through the reticular nucleus. Everything going out have to go. This is like shell covering the thalamus. So everything going in has to go through that. Everything going out has to go through that, and so that kind of like a gate to sense as well as to control the input and output signal from the between thalamus and the cortex. So that's that. So that it is. We didn't really touch on the lateral dorsal. The lateral dorsal is a little bit not fully understood. But other than that, we have a clear understanding about all these the other region. So what's the quiz about? Basically, it's a compare and matching which nuclei are more associated with specific cortical region in the brain. And by knowing the location, we kind of know what their function is about. I want to bring this one here is I want to give you the idea that the cognition is a collective. I think cognition in a way is one term to tell what brain is working. But we need to know that brain function, even though different region has different function, it doesn't work alone. It has a direct connection with the thalamus, and that interaction is critical for that brain area to perform the function. So that's that. So knowing that cognition, the next one is I want to talk about laws of the cognition. And this become quite important because as we are getting older, or the society is getting older, that a lot of people generate the cognitive decline, and the majority or the major type of the cognitive decline, or we call it dementia, is senior dementia that aging population gradually have it, and the major one of that is the Alzheimer's disease. When we look at the Alzheimer's disease, we can look at it in a category of the disease called the neurodegenerative disease. Give me a second, let me. Neurodegenerative disease. So there are common themes among this neurodegenerative disease. Typical neurodegenerative, what does it mean? Neurodegenerative disease refer to a disease that there is a gradual loss of neuron. So if this neuron is in the temporal lobe, that's where we store or process the memory, then it's Alzheimer's disease. If this great loss of neuron in the substantia nigra, then it's a disease of the Parkinson's disease. If these great loss of neuron in the striatum, it becomes Huntington disease. If this grade of loss neuron in the lower motor neuron in the spinal cord, then it's the ALS. So that gradual loss of neuron is a common thing. It's a group of the disease, but when the region of that loss of neuron, where the loss of neuron will determine what kind of the symptom the patient may develop. So that's that. So that is the neurodegenerative disease. So you can imagine that this neurodegenerative disease progress over time. It takes time. It's not acute. It's not happen right away. It happen over time. If we don't find a way to stop it, it will continue going. And very unfortunately, probably all of these diseases that I list here, so far we have no cure. So far we have no way to stop that progression. So that's the common thing of neurodegenerative disease, and that gives you the idea that how do we view the Alzheimer's disease? It's a type of neurodegenerative disease. Another common thing of the neurodegenerative disease is that a lot of these are affected by or influenced by toxic protein, meaning that it's not caused by outside, it's caused by something inside. And so each disease is associated with a specific toxic protein. Alzheimer's disease is associated with this peptide called amyloid beta, as well as tau. This amyloid beta may accumulate to form this plaque, amyloid plaque. The accumulation of tau will form this intracellular fibrillary tangle. So that's Alzheimer's disease. Parkinson's disease is the toxic protein is called the alpha-synuclein. Some of your students, the students has mentioned about this in their presentation, that alpha-synuclein accumulate will form the Lewy body in the cell, and when cell cannot get rid of that, the cell will die, and that cause the gradual loss of neuron. And this happens when this usually happens specifically in the substantia nigra, so we have the dopaminergic neuron gradual loss. Tauopathy is more a general term to describe several diseases. One known disease is frontotemporal dementia, and that is the accumulation of tau. ALS is more complex, but one of the toxic protein is the accumulation of the SOD1. So that's that. So that's another common thing, that in different neurodegenerative disease, we have the gradual loss of neuron. And another common thing among these diseases is that there is specific toxic protein contribute to the progression of that gradual loss of neuron. And that toxic protein can be confirmed when we observe that, right? We can also confirm this by looking at some of these familial case that these disease, some patient has this family history about that. We can see that some of these, when patient has specific mutation to promote the development or the accumulation of this toxic protein, then these patients start to have higher chance or start to develop this disease in the earlier age. So that's another confirmation that not only we observe these toxic protein in the plaque, we also learn that when patient has certain mutation to increase the amount of this toxic protein accumulate in the brain, they also increase or develop this disease at early age. So that confirmed it. Not only that directly mutation to the development of the toxic protein, we also find some related gene mutation or alloy or the variant that associate with the development of this disease. However, because this doesn't directly increase the accumulation of the protein, the relationship of these traits or these gene variants to the disease is not very clear. It's still not very clear. But this can be there is a risk factor. There is a higher risk if individual carry specific type of the allele, yes. So that's that. We are not going to talk all of these. We will talk about the Alzheimer's disease. And this Alzheimer's disease is a major type of the neurodegenerative disease causing dementia in our society. So this one summarized the leading cause of death. I want to show you this one because you can see that Alzheimer's disease is the one of them. This table comes from, I didn't have the age of 2017, right before the pandemic. I think if we do it today, we probably see COVID on the top of this. But either way, Alzheimer's disease is one leading cause of the death. So not just dementia, leading cause death. This was named after the Dr. Alzheimer. That's his patient. And Dr. Alzheimer characterized this disease, not just the dementia, progressive dementia, not just that, okay? He also looking into the brain and to identify there are two pathological hallmarks, including the amyloid plaque, as well as the intracellular neurofibrillary tangle. So at that 100-something years ago, he already identified these pathological hallmarks. At that time, it didn't know what the components in that hallmarks yet. We later learned that neurofibrillary tangle is the accumulation of tau. The amyloid plaque is an accumulation of amyloid beta. And that gave us a sense that, okay, if we can get rid of these, we may prevent or to cure Alzheimer's disease. Still, something that our society is continue pursuing. We don't have the tool yet, but that's part of that provide us the direction to move forward. So that's that. So he identified this. Alzheimer's disease clinical progression starts usually at about 50 years old. And in our it's this major type of the dementia in the older adults. So it's also called the senior dementia. 1% in the age of 60 and above, it becomes 40%. So the prevalence is very high for the age 85 to 89. And that age is not old. A lot of our people live up to this age. 40% of them will have dementia. So this is very critical. This is huge. This will cost a lot of not just the money, but also a lot of our effort about our relationship. So that is a huge thing. Dementia, this Alzheimer's disease progress is a damage in the hippocampus, and that's the region process our memory. So it's influence our memory. When our memory is affected, we lost the experience. We lost our judgment because our judgment is based on our experience. We also affect some other visual spatial orientation, personality. What is personality? Personality is also built based on our experience, our culture, our knowledge, and language. To use the word or to understand language, it's also closely related to the knowledge or memory, the word you learned. Progress a course of 5 to 10 years, and then become disabled, and immobile. So that is a devastating course or journey of this disease. This is what we know. It go through about 10 years. Beginning is a symptom, forget something. We can conduct a course of this neurological test to score the cognitive level. And so here's that mini mental status examination, MMSE. If your score is 27, you're pretty good. If you are near the 25, 26, then that's the early sign of the dementia. You might not get it, the patient might not get it, but if it's continued, then it will get worse gradually. Symptom can be detected, can be diagnosed. Gradually, we have patient will lose the functional independence. In 5-10 years, you will need to stay in a nursing home and then die. So that's what we know, from the early diagnosis to mild to severe. What causes it? The cause, as I mentioned earlier, come from two possible toxic protein. I mean possible because if that is the protein, then we already cure it. So far, all these effort trying to get rid of this are not successful yet. It doesn't mean that this is not a correct target. It just means that we still cannot cure, right? So there are still research going on to trying to cure this disease. But what does this toxic protein? These toxic proteins are amyloid beta and the tau, and they form this neurofibril tangle as well as a plaque. It's kind of shame that even early Alzheimer know it, but it's like 100 years later we still don't have the cure. So what do we know through these 100 years? What do we know about this amyloid plaque, amyloid beta peptide as well as tau? What do we know now? So let's look into it. So what is amyloid beta? This is a peptide, short protein. These peptides are generated from a protein, a larger protein called amyloid precursor protein. These large protein APP are is a membrane, so cell membrane protein, and it will be truncated into smaller peptide. It usually got truncated into two areas called alpha and gamma secretase. This is the enzyme to break it to alpha and the gamma location, and then the produced peptide are soluble, it's harmless. However, in some situation, it may be caught in beta and gamma, and that will produce this toxic A beta peptide. So let's look into that. Amyloid precursor protein, APP, is a membrane protein. It can be caught at alpha and gamma. If it's caught alpha and gamma, these peptides are harmless. However, if it's caught gamma but not alpha, instead of alpha is beta right here, so beta and gamma, these peptides will become toxic. It will get diffused out. This is not soluble. And when it's not soluble, they will tend to accumulate together to form the plaque. So that's that. This A beta is a content about 33 to 43 amino acid. And so the variation of these peptides can have different toxicity. A beta 40 is most abundant, but less toxic. It's not too purely harmless, but less toxic. A beta 42 is most toxic. It's less amount, but most toxic. So that's that. So this one showing you that this is the APP. It's a membrane protein. It can be truncated into alpha and gamma secretase. Then it's harmless. It will be normal. If it's truncated at gamma and beta, then this A beta amyloid beta peptide will be diffused. And because they are not water soluble, they will accumulate it. They form the oligomer aggregate. That means it's smaller oligomer, or gradually become like large plaque. A beta itself, the original idea about this A beta is that it's an important membrane protein, is in the neuron. It's important to regulate our synaptic formation. And and the truncation of it plays an important role for neuroplasticity, synaptic formation, or the extraction. Alpha secretase will make it normal. Beta secretase will produce A beta. A beta can be cleared away, but if our clear system doesn't work very well, then it accumulates. This also related to the APP, this amyloid precursor protein, membrane trafficking or transportation. Just like every other protein synthesis from DNA to RNA, RNA to protein. This RNA to protein, this translation process occur in the RAF ER, in the ribosome. Ribosome does it, then it will move into the Golgi apparatus, still anchored in the membrane, then to wrapped, to packed in the vesicle. So this is APP. This APP's function is to regulate our synapses, so it will be transported into the dendrites where we have the synapse. In the dendrites, then it will be moved into the plasma membrane, and then it will also get engulfed, endocytosed. When they are in the outer portion in the membrane, it can be truncated more easily by alpha secretase, so that will be harmless. However, when they got engulfed, the pH level in the endocytosis in the vesicles are different, and the protein are more likely to be truncated by the beta secretase. It can be recycled, engulfed, used again, or be lysosome get degraded. However, through this endocytosis, through this process, it increases the chance to be truncated by the beta secretase, and that increases chance to be to generate the amyloid beta. And so you can imagine that these amyloid beta will be generated more and more when we are aging, and when we are young, it can be cleared away or be digested, but when we are getting older, then these peptides may accumulate more and more. So just showing you, this one just showing you that when it's in endocytosed, it has higher chance to be truncated by beta secretase. And that's how our cell generate the A beta. So we know how it's generated. We know a lot through these 100 years. We know that is produced from the amyloid precursor protein. Maybe the quiz question could be that which protein is the precursor for the amyloid beta? It's APP, amyloid precursor protein. And how is that truncated? Gamma secretase and the beta secretase. Yeah, so that's that. So when it's produced, it will be accumulated, and over time, it accumulated, it become a plaque. And when it becomes a plaque, it becomes harder to get rid of. If it's single one, it can be removed through our cerebral spinal fluid, and to clear away, to move it into the plasma and to get rid of it. Yes, into the vein, into the plasma, get rid of it. Get rid of it from our brain. One thing that we also know already is that clear system is also associated with sleeping. You know, another good thing about sleep. So that's why, you know, don't cut your sleep don't sacrifice your sleep for something else. If you need to study, you know, you need to sacrifice your movie time or TV time or something else, but don't sacrifice your sleep, because sleep is the time that this clear system can be accelerated, can be faster. A beta itself is toxic. This paper summarized non-toxic function of the A beta. It caused the mitochondria damage, increase the oxidative stress. That's also oxidative rate stress, and it's damage to neuron, increased amount of calcium that actually can induce the apoptosis. It also causes damage to the DNA. It also causes damage to the synapses. When the synapse is injured, the neural connection is disconnected, and basically that's how our cognitive decline, because all this connection is not connected very well. So that's the A beta toxicity. So that summarizes it. A beta can cause mitochondrial damage, increase the oxidative stress, and also cell membrane can be disrupted by the A beta aggregates. The DNA can be damaged by causing the telomerase inhibition, can also affect the synapse. So that's A beta. So A beta is toxic already. Another thing that A beta also do is to activate a peptide or enzyme called protein kinase. What does kinase activity? What does protein kinase do? Protein kinase, our body, our cell has about 100 or 1,000 type of protein kinase, maybe hundreds, not thousands. A lot of protein kinase, different type of protein kinase. Protein kinase do phosphorylation. So you can see that protein can be decorated by adding additional phosphate to it. One, not just protein, DNA can be, not DNA, histone can be phosphorylated, chromosome can be phosphorylated, ADP can be phosphorylated, ADP to ATP, so that's phosphorylation. And that phosphorylation requires an enzyme, and that enzyme is collectively called the protein kinase. A beta can trigger a specific type of protein kinase to cause phosphorylation to tau. So that links these two together. We have the A beta accumulation as well as tau accumulation. Tau accumulation is achieved through first tau phosphorylation, hyperphosphorylation in fact. So A beta can activate protein kinase then cause tau phosphorylation. Then the question is, what is tau? Tau is a microtubule associated protein. Tau is microtubule associated protein. What is microtubule? Microtubule is a type of cytoskeleton. We know it already, right? Microtubule type of cytoskeleton is from microtubule. It's formed by tubulin. So these are the tubulin. This is very important in the exon from the exon transport. So this is exon transport. This is vesicle carried by the motor protein to move this vesicle up and down, right? Or like anterior or retrograde transport. These microtubule are stabilized by tau. So this is tau stabilized the microtubule. Tau stabilized the microtubule. When tau is hyperphosphorylated, so this does refer to that phosphorylation. Hyperphosphorylate, too much of phosphorylation by amyloid beta trigger this protein kinase. Then tau become detached from the microtubule. When tau is detached from the microtubule become unstable. So microtubule will be degraded. You need to know that microtubule are produced by the assembling of the tubulin. So tubulin can be easily just spread away, and microtubule will be disassembled. And that will cause microtubule degeneration. Cell will be injured. So that's how tau. And then tau can be accumulated to form neurofibril tangle. So that's that. So that's A beta and tau. Alzheimer's disease, 90% are sporadic. 10% are familiar. These familiar forms are associated with the earlier thing that we talk about this one here. The APP mutation tend to be more APP will be more A beta. Presignaling 1, presignaling 2, these are the beta and the gamma secretase, so that increase the cutting of the APP into A beta. So that is the familiar form. Only 10%, about 10% of the AD is familiar. 90% are sporadic. Familiar one also earlier Alzheimer, that patient will develop Alzheimer at a younger age. Sporadic one is more like aging one, senior one. Another risk factor is apolipoprotein E, apolipoprotein R. The apolipoprotein R, the protein to carry the lipid in the blood. There are different types of apolipoprotein E. 2-alloy is alloy 4, and if the person has both of allele 4, then it has a significantly higher chance to develop the Alzheimer's disease. The Alzheimer's disease can be detected by using imaging like MRI, and the one thing to do is to look into the temporal lobe, especially the medial temporal lobe. That's where we have the hippocampus. So we can see that these regions can show shrinkage, showing that the neuronal loss. This medial temporal lobe right here, if you do the sagittal view through the medial plane, you can see that this medial temporal lobe right here, this temporal lobe, this is medial temporal lobe, include the hippocampus, including the entorhinal cortex, perihinal cortex. So these are the parahippocampus. So these are the region very important for the memory formation of memory processing. And if the neuronal loss in this region, we have the memory loss, and that is the earlier sign of the Alzheimer's disease. We can also do imaging on the plaque. So we can inject the dye or the marker to bind on the plaque, and this dye shine the radiation So we can use position emission tomography, PET. After the injection, we can see the distribution of this plaque in the brain. And so that can show you that plaque non-invasively. We can also do spinal cord, the lumbar puncture to get the CSF, cerebral spinal fluid, and we will detect the level of the tau as well as the A beta. So A beta is constantly get generated in the brain, but it can clear away. We can clear it into the CSF. However, if it get accumulated in the brain, we will see a reduced A beta in the CSF. On the other hand, that A beta in the brain can trigger the tau phosphorylation, so we will see more tau in the CSF. So by doing the lumbar puncture, getting the sample of the CSF, we will see increased tau, phosphorylated tau, and a reduced A beta. And so that's what we know at this point. So this is a paper in 2016, kind of like not exactly very new, but kind of what we know now. Earlier, and we know a lot. We know how A beta is formed. We know the relationship between A beta and the tau. We also developed several tools to conduct early diagnosis. Before we need to diagnose it by doing the neurological test and the patient showing loss of memory as a sign of the early Alzheimer's disease. But now we can detect it way early. So this is the dementia. This is the neurological performance, whether that's normal, mild cognitive impairment or dementia. But now we can detect way early. So, for example, this blue line indicate the CSF. So by doing the lumbar puncture CSF, we can detect it way early. We can also perform the PET image of the amyloid plaque. We can also detect the CSF tau. We can also use MRI to detect the shrinkage of the temporal lobe, medial temporal lobe. And that is way before our neurological performance to show a decline of the memory. The problem of all this is that nobody will do this test unless it shows or unless he or she experience the decline of the memory. Nobody will just regularly do this test. Nobody will do that. And so that is another issue, that we cannot early detect this disease. And when we already know, it kind of already passed through this critical window for the treatment. So that is an important issue, that how are we going to treat or cure this disease. So the treatment, currently there is no cure, but there are drugs to help with the symptom. Two FDA approved drugs there are new one. I didn't put it here. I think one just approved last year, 2021, this year. It's kind of, excuse me. It kind of arguably passed. I think when FDA review when FDA 等一下 hold on a second. So two currently commonly used drug. I know there's new one, but two commonly used drug is ACE inhibitor and the NMDA antagonist. So ACE inhibitor work on the cholinesterase inhibitor. So this one basically, this cholinesterase is the enzyme to get rid of the acetylcholine in the synaptic area, and that inhibition can keep this acetylcholine stay in the synapse for longer time, and so that can maintain. Acetylcholine is important to in several regions, particularly for neuron in the memory, in the hippocampal area. So that even though neuron continuing dying, but the acetylcholine can this drug can maintain the acetylcholine in that region, and so allow these neurons to perform better. Another one is the NMDA antagonist. This one is to block the NMDA receptor, as we know earlier. That is the glutamate receptor. And sometimes glutamate receptor cause glutamate excitotoxicity is one mechanism to cause neuron loss. So this memantine can be subscribed for patients to prevent or to reduce the excitotoxicity to the neuron. So that's these two. I am working, okay? All right. So this is some other idea. Okay, so this is just cholinesterase that showing you that acetylcholine is truncated. So it will keep the acetylcholine more, in the cliffs for longer time. There are many ideas are currently working on to find way to treat Alzheimer's disease. So this is paper in 2019, and showing you that currently there are about 2,000 some clinical trials to try to find the cure of treatment for the Alzheimer. And as you can see, the traditional amyloid hypothesis, tau hypothesis, only take about maybe 1/3 of this trial. And several other ideas are currently being tested, including the virus idea, diabetes idea. There is connection between the diabetes and the Alzheimer's disease. Inflammation idea, that the inflammatory response can also participate in the neuronal loss in the Alzheimer's patient. Exercise idea, we will talk about that. This is a good one. It's free in a way. You just need to do exercise and it actually works. Neurovascular idea and mitochondria damage idea. So there are several ideas currently working on. Question is, what can you do, right? What can you do? You are young, but we all will get old. What will you do? One thing I highly recommend you is, or is already known, is the physical activity. This is, you need to know that people try a lot of things. You probably heard that there are certain food that can improve memory, right? Fish oil, avocado, or some nuts or something like that to improve memory, improve brain function. Red wine, what else? So there are different kind of idea, but all has been tested, not working. One thing work is exercise. So this is the original paper several are two. These one is the initial paper, the very original paper. I think that's this one here. Original paper talk about exercise, and what people found is that when people exercise more than 3 times a week compared to less than 3 times a week, can significantly reduce the risk to develop dementia. So I think this will go along with the several other like mechanisms such as neurovascular, cardiovascular system. Exercise, maintain your cardiovascular health, and that can help with the reduce of dementia. Exercise can also increase the neurogenesis in a way that can, build up your cognitive reserve. Yeah, so reserve, yes. So in order to, when there's any decline, you can have more room to sustain with that decline. Exercise can also help with the diabetes condition. You can better utilize the energy, the glucose, and the lipid in the body. So that also can help with preventing the development of Alzheimer's disease. I think so. The effort from the exercise is different. It has different like a pathway to help with the development of or the prevention of the Alzheimer's disease. So that's that. That's it. Let me know if you have any questions.